Hi, this is Josh, Dream Body Clinic, and today we're talking about Muse cells. In this video, you're going to see Dr. Dezawa, the discoverer of Muse cells, explain how they work. So what she shows is they basically act like a macrophage. Now, a macrophage is an immune cell that's job is to go in. Basically, it'll find damaged cells or debris, it'll encompass them, absorb them, and eliminate them from the system. It's like the cleanup crew. So the Muse cells act like that at first. They seek out inflammation or damage. In the video you'll see here, she's showing some apoptic liver disease that they went into the liver. Now, this is just from an IV. The Muse cells went through the system, found the damage at the liver, and they started consuming the bad cells. And then instead of just getting rid of them, the cool part is they then differentiate into liver cells. And they can do this with any type of cell in the body, with skin cells, with vascular cells, with organ cells, you know, the huge list of different things that they can do. Pretty much any cell in your body, they can do this. And the cool part is they're non-tumorigenic, so they have no risk of turning into tumors or cancer. And they'll simply replace bad or damaged tissue with new, fully functional, healthy cells. It's really incredible. So stay tuned, watch, let Dr. Dezawa explain it in her own words here in this video. We flew her in from Japan so she could explain to our team how these work, and we are working directly with her to offer her Muse cells now. You can check that out at www.dbcmusecells.com.
Now, I would like to show you first how Muse cells are similar to macrophages. So green is human Muse cells collected from bone marrow. The red is mouse apoptotic liver fragments. So within 24 hours, the Muse cell capture and eat, right? And by 24 hours, if we take out only the red signals that were incorporated in the cytoplasm of the Muse cell, you can see that Muse cells are really great eater. They're all the time hungry. They're hungry. So you now realize that Muse cells aggressively capture the dead cell fragment and then they will eat it, okay? Now, like this manner, the difference, this is very big difference between Muse cell versus the macrophages. So macrophages, they eat, they digest, and bye-bye, right? They're for cleaning company, but the Muse cells are different. After eating each dead cell fragment from cardiomyocyte, neurons, hepatocyte, or whatever, or vascular cells, skin cells, keratinocyte, whatever. They capture the signal, and then they differentiate into the same cell type, and then they replace. So I will just skip the detailed molecular mechanism, but this was published 3 years ago in Cell Molecular Life Science. That once Muse cell capture the fragment from the differentiated cell, cell fragment, like apoptotic bodies, this is, this part. This is the fragment, okay, here. So the fragment from that cell fragment, they contain the DNA, RNA, even organella like a mitochondria. The most important thing is transcription factors. This is protein. And then they will fuse with the Muse cell, the cell membrane. And then within maybe 5 or 6 hours or more quickly, the content will be quickly released into the Muse cell cytoplasm, and the transcription factor translocate into the Muse cell nucleus, and then they sit in the right position to start differentiation.
Yeah.
So Muse cell finish differentiation within 2 or 3 days. It's very quick.